Hi, everyone. For those of you who may not know, my name is Sananda, and today I will be presenting information gathered from a research article titled Circulating Tumor Cells with Metastasis Initiating Components Survive Fluid Shear Stress During Hematogenous Dissemination through CXCR4 PI3K/AKT signaling. So, some information to recap on before we dive into this paper is an overview of metastasis. Metastasis is the process by which cancer spreads from a primary tumor to other parts of the body, and it's unfortunately responsible for most cancer related deaths. The seven steps of the metastatic cascade are invasion of surrounding tissues, intravasation, which is entry of cancer cells into the lymphatic or the blood vessels, transportation within the circulatory system, arrest in the microvessels of various organs, extravasation, which is exit from the lymph or blood vessels into tissues, the formation of micrometastasis, and colonization, which is when micrometastasis turns into a macrometastasis. So, why is circulation so difficult for cancer cells? Well, cancer cells face many challenges when circulating in the blood vessels. One of these challenges is anoikis, which is cell death associated to a lack of attachment to a solid substrate. They can also be killed by immune cells that are present in the blood vessels. But one particular challenge that we will talk a lot today about is the physical stress, specifically fluid shear stress that cancer cells go through when in the blood vessels. What allows some cancer cells to survive versus others? It's not well understood why some cells survive and others don't. But that leads us to the purpose of the study. This study aimed to find out why some circulating tumor cells survive when exposed to fluid shear stress, which is the force generated by blood flow, and whether these surviving cells have enhanced metastatic potential. Some of the research questions are why some cells resist death due to shear stress, what pathways allow the survival, and more importantly, do these cells contribute more effectively to metastasis? Now, onto the methods. Firstly, the scientists modeled cancer cell survival in the bloodstream. They did this by stimulating blood flow conditions and placing the cancer cells in this simulation. Then they would take the cells that survived. These cells are called shear stress selected metastasis initiating cells, also known as SSMICs, which is what we will refer to them in this presentation. They isolated these cells and then compared them to control tumor cells and suspension tumor cells. This was to prove that differences between tumor cells were caused by shear stress specifically. They then performed many functional assays to test metastatic ability. The adhesion to endothelial cell assay tests how well cells can stick to the endothelium of the blood vessel. This is important for exiting the bloodstream. The trans-endothelial migration assay tests how well cells pass through the blood vessels, which is also required for metastasis. The migration and invasion assay also tests how cells move and invade tissues, which can be a measure of how aggressive the cancer is. This study focuses on CXCR4, a receptor involved in regulating cell migration, proliferation, and survival. The study also focuses on the PI3K/AKT pathway, which is a survival signaling pathway that, when activated, causes cells to avoid cell death. Upon activation, CXCR4 can stimulate the PI3K/AKT pathway, helping cells to survive and resist apoptosis. This is especially important under shear stress conditions where cells would otherwise undergo cell death. They also used mouse models to determine whether these cells could actually form tumors and metastases in vivo, which is in a real life model. So, onto the results of this study. As you can see, 1b, figure 1b shows that SSMICs show an increased attachment to the endothelial layer. You can see this by an increase in red dots compared to the control and the suspension at all the time periods, 1 hour, 2 hour, 4 hour, and 8 hours. Figure 1c shows that SSMICs are also better at moving through the endothelial layer. You can see this by an increase in transmigrated cells or SSMICs when compared to control and suspension cells. This suggests that these cells are better at initiating metastasis by attaching to and crossing blood vessels. Figure 1d shows that SSMICs had an increased number of migrated cells. Again, you can see this by SSMICs having a significantly higher number than the suspension and the control cells. Figure 1f shows that SSMICs also form more colonies. Again, you can see this by it having a significantly higher number than suspension and control cells. This indicates that these cells not only move more effectively, but also have a greater ability to grow and form new tumor colonies. In mouse models, figure 2a and 2b show comparisons between the groups in tumor weight and volume. It shows that SSMICs and tumor-inducing cells, TICs, had similar tumor weights and volumes, and that both of these groups are significantly higher than control and suspension groups. This suggests that SSMICs have tumor-forming capabilities comparable to tumor-inducing cells, TICs, which, as we know, is a highly aggressive cancer cell that is capable of forming many tumors. Next, they examined CXCR4 and found that its expression was significantly increased in SSMICs. We can see this as CXCR4 is the row in red in figure A. And as you can see, there is more of a red line through SSMICs than there would be for suspension and controls. This suggests that CXCR4 may play a role in helping these cells survive and become metastases and become metastatic. To test this, they used AMD 3100, which is an inhibitor of CXCR4. When CXCR4 was blocked, migration was reduced. So we can see this in figure 4H. 0 represents that there was no exposure to shear stress, which is shear stress. And then 20 represents that there was exposure to shear stress. And as we can see, when after exposure to shear stress, the controls were much higher than the cells stimulated with AMD. This shows that there was less migration when CXCR4 was blocked and inhibited, which represents that CXCR4 is important for these metastatic behaviors like migration. In figure 5C, the researchers compared cells with and without CXCR4 under shear stress. CXCR4 minus is without CXCR4 and CXCR4 plus is with CXCR4. They found that for both types of cells, there was an increased migration under shear stress. We can see this as with the CXCR4 minus at 20 compared to 0 there was an increase. And with CXCR4 plus from 0 to 20 there was also an increase. However, the increase was much greater in CXCR4 plus cells compared to CXCR4 minus cells, which suggests that while shear stress alone can enhance migration to some extent, CXCR4 significantly multiplies this effect. And the smaller increase in that's seen in CXCR4 minus cells could indicate that other pathways may contribute to this, meaning that while CXCR4 is a major mechanism, it is not the only mechanism involved. Figure 5e presents the results of a Western blot. Darker bands means there's more protein. PPI3K is the activated form of PI3K. As we can see, the SSMICs have more PPI3K, indicating that this pathway is more active. Li plus 20 is PI3K's inhibitor. Figure 5G and 5H show the results of PI3K inhibition, and it shows that both migration and colony formation decreased when the inhibitor was present. This means that blocking PI3K reduces metastatic traits and shows that this pathway is necessary for metastatic behavior. They then knocked down CXCR4 using silencing RNA, and they found that this also led to reduced migration and colony formation. Again, reinforcing that CXCR4 is vital to metastasis and invasion. In figures 5J and 5K, they compared CXCR4 knockdown with CXCR4 knockdown with PI3K activator. So one group had CXCR4 just knockdown, and then the other group had CXCR4 knockdown, but it had a PI3K activator. And the results showed that while CXCR4 knockdown did increase migration and colonies slightly, there was a larger increase of migration and colonies with the PI3K activator. This shows that PI3K acts downstream of CXCR4, as if it acted upstream, then PI3K would have had no effect because CXCR4 would still be, there would still be no CXCR4. But the results of this showed that PI3K acts downstream of CXCR4. Now, what does this tell us? This study shows that fluid shear stress acts as a selection pressure, allowing only a small group of tumor cells, called SSMICs, to survive. These cells are not only resistant to apoptosis, but they also have enhanced metastatic abilities, such as increased migration and colony formation, and which also allow them to form tumors efficiently. CXCR4 promotes these effects by activating the PI3K survival pathway, which allows cells to resist apoptosis. The findings of this study show that targeting CXCR4 or PI3K signaling can reduce the metastatic traits of these cells. This is clinically important because metastasis is the main cause of cancer-related death, and this study identifies CXCR4 as a key factor that allows cancer cells to survive in the bloodstream. So targeting CXCR4, either by using inhibitors like AMD 3100 or silencing it using silencing RNA, after targeting CXCR4, it may be possible to reduce metastasis and improve patient outcomes, although further research is needed to confirm this. However, CXCR4, if you measure CXCR4 cells in the patient blood, you could also detect the risk level of metastatic cancer. However, some limitations of this study is that in vitro shear stress is not equivalent to full human conditions. So the results may not correlate in humans as we expect it to. Also, mouse models may not fully translate to humans. Further testing is needed in models to see if CXCR4 inhibitors would work how these scientists predict would. And as mentioned previously, there are other signaling pathways involved that we need to identify and do further research on to gain a holistic picture and figure out how to best treat metastatic disease. Thank you for listening. I hope this was informative.